First, John, you're being very modest when you presented your Vericel experience. Vericel was the first to have a rapid sterility test using the BacT-Alert for lot release for Carticel in 2004, as presented in this 2005 cytotherapy publication. And they had the first rapid Mycoplasma test using real-time PCR approved in 2013. I compliment Vericel in publishing and sharing their experience so everyone can benefit from lessons learned. So thank you, John, for your presentation today, presenting the 5-year commercial experience by Vericel for nucleic acid testing, for Mycoplasma testing. I also want to echo John, in that the USP proposed Mycoplasma nucleic acid test, General Chapter 77, is currently published in the Pharmacopoeia Forum, and the public comment period ends at the end of this month. So it ends November 30th. So all you SMEs out there, please read the chapter and submit your comments. I will be presenting on the BioFire Mycoplasma nucleic acid test method that provides sample to result in less than an hour. I included cell therapy and bioproduction in my title because I will be providing data supporting both markets. Before jumping right into BioFire next generation PCR, because I'm a microbial ecologist, I always like to throw evolution somewhere in my presentation. So I want to illustrate the evolution of nucleic acid testing. And I'm showing here conventional PCR as real-time PCR. The pro is that you get results quickly. For Mycoplasma testing, it typically takes about 5 hours, and that's what John also presented, that it takes about 5 hours. However, there are a few pain points to conventional PCR, such as that it does require a dedicated lab with extensive hands-on and usually a highly trained molecular biologist. These systems are prone to pipetting errors, and it's an open system. So in cases where you need that excellent chain of identity and chain of custody, especially in cell and gene therapy, it's best to use single-use closed systems. Nucleic acid testing has evolved in the past decade to include sample to answer systems that are fully automated. There are many benefits, excuse me, of sample to answer systems, including a much faster turnaround time with reduced hands-on time and required expertise. They are robust, closed, single-use systems that reduce contamination risk and human error. Now, typically, these instruments have a small footprint, and this allows the location to be flexible, and the results are automatically interpreted by the software. From a business perspective, there is a benefit for labs that currently outsource testing by allowing Mycoplasma testing to be performed in-house. So this eliminates costly outsourced testing and allows you to respond and recover from a contamination event faster. Now, for labs that are currently using traditional PCR, there is a benefit of increased lab efficiency. The ease of use streamlines workflows and reduces subjectivity, human error, and retesting. John presented the impact of invalid results for their Macy product. Fewer investigations will definitely optimize the process, and that will ultimately impact the patient outcome. There are several commercially available sample to answer nucleic acid systems. This isn't new. They've been around for over a decade, and they've been used in clinical diagnostics, public health, and biodefense. But this ease of use approach has only been recently adapted for pharmaceutical applications when bioMérieux launched its BioFire Mycoplasma test system in 2020. Next generation sample to answer has been a proven technology in use in clinical settings for bioMérieux for over 10 years. There are 7 BioFire panel clinical applications, and globally, there are over 20,000 BioFire modules running clinical pouches. So the clinical knowledge over the past decade was used to develop the BioFire Mycoplasma test system that provides results in about an hour. Now, our system was designed to allow easy, rapid at-line testing that can be performed by anyone, anywhere, anytime. And this testing can be performed on raw materials, in process, and final product. The BioFire Mycoplasma test can be used for in-process controls. There's no specialized lab or molecular biology skills that are needed. There's a minimum hands-on time with results in less than an hour. The small footprint does allow it to be moved out of the lab and closer to the production floor. And this allows testing to be performed on demand anytime to check the quality of your manufacturing. And in-process control testing will allow for early contamination alert, and this protects your process. The test can also be used for release testing. BioFire Mycoplasma can detect over 130 strains and provides automation in 21 CFR Part 11 capable software and increases data integrity. It's comprehensive and validated and meets regulatory requirements for release testing. The system consists of just two components, the film array instrument and the single-use molecular lab in a pouch disposable. So with only these two items, you have everything you need for fast, accurate, and simple mycoplasma testing. Here are some pictures of the actual machine and reagent. The system comes standard with 2 film array units for increased throughput and redundancy, but up to 8 film array units can be added to a single computer, which would allow for batch testing. The disposables are packed 6 to a box and are stable at room temperature. Now I'm going to go into a little bit more detail of each component. I'm going to start with the instrument. The film array instrument has a small footprint. It's the size of about it's the size of a small shoebox. And despite the small footprint, the system is able to perform the extraction, amplification, and detection that typically requires several larger instruments. Now, as unique as the film array instrument is, I would say the real magic is in the single-use disposable. It's about the size of an index card that we call a molecular lab in a pouch. The pouch contains all of the reagents that are required to run the extractions and multiple types of PCR. And these reactions and detection occur in the fluidic circuit at the bottom of the pouch. I'm now going to go into detail about each of these steps. So after hydration and the addition of the sample to the pouch, the extraction process begins. The silica and magnetic beads are used for nucleic acid extraction and purification. After the purification step, the first stage PCR includes reverse transcription with a multiplex PCR. And because we're utilizing both RNA and DNA, the presence of live mycoplasma in the sample enhances the sensitivity of detection. The second stage of PCR is a nested PCR. Each well of the array is a single PCR, and we use a total of 14 unique PCRs in triplicate, targeting 3 different genes to detect more than 130 species of mycoplasma. There are several controls that are integrated into the pouch to ensure quality of the results. So as you can see here, the entire process is monitored from extractions through PCR2 with a freeze-dried yeast. Yeast RNA is used to check the reverse transcription as well as PCR1. And finally, PCR2 is checked by DNA fragment controls. For additional controls, such as those needed for incoming QC or training or qualifications, we have developed an external positive control kit that utilizes synthetic modified DNA mix. So the modifications shift the temperature of the melt curves so that they can be distinguished from natural mycoplasma DNA. So now let's talk about the workflow, software, and results. This slide compares conventional real-time PCR to the BioFire sample to answer PCR method. Conventional PCR might offer speed, but not ease of use. The BioFire method gives you ease of use in about 1/5 the time of conventional PCR. Now I have a short video that will walk you through the steps of the pouch workflow.
The science happens in the single-use consumable we call a molecular lab in a pouch. The pouch contains all of the reagents required to run the extractions and multiple types of PCR reactions. Preparing the sample is simple. The technician loads the pouch into the loading block. Hydration solution is injected into the pouch through the blue inlet port. The sample is added to the sample injection vial using the transfer pipette. Sample buffer is added to the sample injection vial. The lid of the sample injection vial is closed, and the vial is inverted 3 times to mix the sample. The sample buffer mixture is injected into the pouch through the red inlet port. The pouch can now be loaded into the film array instrument.
So it is that simple. We have validated 2 off-the-shelf protocols for testing. The first is intended for in-process controls, and it's a simple addition of 0.2 ml of sample to the pouch. And this protocol takes roughly 2 minutes of hands-on time. I will discuss the limits of detection a little bit later when I get to the validation data, but this 0.2 ml protocol has an LOD of around 30 CFU per mil. The second off-the-shelf protocol takes 10 mils of sample and concentrates it down to achieve the 10 CFU per mil required for final release testing. So with double centrifugation, the total hands-on time is about 25 minutes. Now, since launching the system in 2020, we've had a lot of interest in testing cell and gene therapy sample types, and we have evaluated a single centrifugation protocol using Jurkat cells that allows testing samples with cells that is suitable for release testing. What we're experiencing, especially for cell and gene therapy, is the need for protocol flexibility. And we're seeing great results with BioFire Mycoplasma to satisfy this need. After you set up the pouch, you start the run through the intuitive software. So despite the simplicity of the software, it has been specifically developed to meet pharma standards and provides 21 CFR Part 11 compliance. It includes barcode scanning and automation to reduce human error and increase data integrity. Like everything else with the system, the reports are also simple and straightforward. You can see here that the results are simply mycoplasma detected or mycoplasma not detected. Additionally, the reports indicate whether or not the internal controls passed. So now let's take a look at some data. We have used the International Pharmacopoeia guidelines to validate the BioFire method using the criteria described in the major chapters for mycoplasma testing, such as LOD and organism type. We've also used the various chapters describing validation of alternative methods for qualitative tests, and these require the parameters of limit of detection, specificity, robustness, repeatability, and ruggedness. These detailed primary validation data are contained in our validation report that is available to users. Today, I'm just going to show a very high-level summary of this data. The organisms chosen for validation were based on the requirements from the 3 major pharmacopoeias and are listed here. We determined the LOD of both the 0.2 ml and 10 ml protocols. For the 10 ml protocol, you can see that all of the organisms tested were detected at less than 10 CFU per mil, with the exception of a Mycoplasma salivarium, which was at the limit of 10 CFU per mil. So this meets the criteria to replace the compendial cell culture method. The 0.2 mil protocol, we were also able to meet this criteria for 5 of the 10 organisms tested, but all were detected at 33 CFU per mil. We also tested the LOD of the shown microorganisms with CHO cells at a concentration of 5 times 10 to the 6 per mil, and there was no negative effect. For specificity, we performed inclusivity testing that tested additional strains, as well as performing an in silico analysis that shows a total of 132 Mycoplasma species detected. For exclusivity testing, we tested closely related bacteria, and none were detected at concentrations of less than or equal to 10 to the 6 CFU per mil. For interference, we tested a wide range of potentially interfering substances related to biopharma manufacturing at the concentrations shown, and none demonstrated interference with the exception of 1% bleach, but that was to be expected. Since the launch in 2020, numerous internal studies have been performed on cell and gene therapy samples. All of the listed matrices here were found compatible with the system. I think it's worth mentioning again that though it's not part of our ruggedness, robustness, and repeatability data, the BioFire system is only new for pharma applications. With 510K clearance by the FDA, there are over 20,000 BioFire modules that are routinely used for clinical syndromic diagnosis of disease and infections. Again, all of the details used to generate the summary data I've shown today are available as part of our comprehensive implementation package, which includes on-site support. Excuse me. We officially launched in the US in May of 2020, and prior to the launch, we did work with customers in the US and Europe to perform some beta testing. I would like to share some of that data today. The first 2 companies are BioProduction, and the last 2 companies are Cell and Gene Therapy. The first company tested the 0.2 mil protocol for in-process controls. They performed interference studies on their used or spent culture media from their bioreactor, as well as their CHO bulk product. They also performed inoculation studies with 3 strains at 2 concentrations in CHO cells. So as you can see here, the data show that there was no interference with their spent bioreactor media or their CHO cell bulk product. There were no false positive or results that were invalid. The inoculation studies in CHO cells were aligned with our internal data. There was 100% detection for samples that were inoculated with greater than 10 CFU per mil, and between 50 to 70% detection for samples inoculated with around 10 CFU per mil. The second company tested the 10 ml release protocol using CHO cells that produce monoclonal antibodies. They tested interference at different CHO cell densities and inoculated with 7 different strains at concentrations ranging from 10 down to 0.1 CFU per mil. You can see here that there was no interference. There were no false positive results that were detected at low or high density for all 63 tests performed. The spiking recovery studies were aligned with our internal data regarding LOD for the 10 ml protocol. Additionally, the LOD of the BioFire method was consistent with this company's current in-house PCR method. Shifting to the cell therapy evaluations, company A tested CAR T cells at a concentration of 3 times 10 to the 6 cells, and company B tested an autologous cellularized scaffold that contains 2.5 times 10 to the 4 cells per mil. Company A evaluated the 0.2 mil direct test protocol, as well as a 0.5 and 1 mil centrifugation protocol, while company B tested a 10 mil single high-speed centrifugation protocol. So that shows some flexibility in the methods and protocols used. Here are the Mycoplasma species tested by each company. This table summarizes the results for the direct test 0.2 mil protocol. 3 out of the 3 CAR T samples inoculated with a target of roughly 40 CFUs per mil of Mycoplasma pneumoniae were detected. For the 10 mil release test protocol, company A used a 0.5 mil product sample with a 2-step centrifugation, while company B used a single high-speed centrifugation. And for the release test, the results showed 100% detection for both sample types inoculated with between 1 to 40 CFU per mil of the tested mycoplasma. For the release test, company A reported an LOD of less than 10 CFU per mil, and company B reported an LOD of around 1 CFU per mil. And lastly, there were no false positive or false negative results reported by either company. Comparing the 3 methods, the BioFire Mycoplasma solution provides sample to answer in less than an hour with minimum hands-on time or expertise that's needed. Since it's a single-use enclosed system, the risk for human error contamination is low. The reagents are room temperature storage, and it allows for at-line testing. The benefits of implementing an in-process rapid contamination test, such as the BioFire Mycoplasma, is that it allows for early contamination alert to protect your process. It's easy to use, and it can be brought out of the lab to the shop floor where it's needed. And lastly, it is comprehensive and validated to meet regulatory requirements. I just want to add that in addition to mycoplasma testing, bioMérieux has a wide portfolio of smart quality control solutions that optimize productivity. With that, I complete my slide decks. I would like to give a special acknowledgement to my colleagues, Will Berry and Jeremy Robertson, who helped me with much of the content that I presented today. And if you have any questions, please feel free to contact me. And with that, I will give back the screen to you, Marcus.